there's a particular subset of the regulatory T cells known as the T follicular regulatory cells, which essentially has this specialized function for the control of B cell antibody production. And of course, while we want antibody production in many contexts, because of course we need pathogen-specific antibodies to help protect us, left completely kind of unregulated, when you lose the kind of the controlling element of the Tregs and the TFR, what you essentially see is an expansion of your kind of autoreactive antibodies, so your autoantibodies. And in many kind of autoimmune diseases, and also more recently, we find they're produced in severe infections such as COVID-19. These autoantibodies can attack your own body, which of course is something we'd like to avoid. So to be able to see all these things at one time, we tend to use what's called the kind of single cell technologies. So my lab uses kind of mass cytometry. It's probably our main kind of workhorse for a lot of work. And part of the reason we tend to favor this particular technology is it's got a decent balance between high sensitivity and resolution, being able to measure in the range of kind of up to 50, 60 markers at one time, and also being able to look at like large numbers of samples and millions of cells. Which is particularly important when you're interested in, relatively rare subsets such as regulatory T cells, as I am. And so what we're looking at here is is a detailed look at the regulatory T cells in human blood, because regulatory T cells, despite being relatively rare, do have their own kind of variety and heterogeneity within them. And part of the reason we're interested in this population is when we look in our cohorts of severe COVID-19 bacterial sepsis, we see that this population, which we call the pre-TFR, kind of precursor TFR, they're very much kind of suppressed in terms of frequency in the blood of patients with severe COVID-19 and bacterial sepsis. And essentially here we're looking, down on the x-axis, we can see our kind of longitudinal cohort of intensive care patients. And you can see that in most of those time points, they have reduced frequencies of these TFR. But essentially in this work, we were able to demonstrate that, we can see this kind of precursor population of TFR, which has kind of, early TFR kind of characteristics, and if stimulated will kind of turn into more mature TFR. And the loss of this population is associated with the increase of autoantibodies and also kind of activated atypical B cell populations, which tend to be our autoantibody producing B cell types. And so essentially this led us to just making this kind of like, again, kind of CyTOF single cell readout Treg suppression assay system. And we made kind of a single cell mass cytometry readout, which gave us the ability to see both kind of phenotypic markers of activation status, kind of cell typing and lineage markers, and also other things such as proliferation tracking via CFSE, which you can adapt into a kind of CyTOF context, and also measurement of cell cycle, DNA, RNA, protein production, and the such like, all at the same time. And so this is just a direct demonstration of the Treg's ability to both inhibit the proliferation of activated B cells and inhibit their differentiation into these kind of atypical phenotypes, which is something we see suggested in the blood ratios, but obviously this is a more direct demonstration.